restoring aquatic biodiversity, okay? Restoring, active verb, present tense, always. And that's probably the way it will always be. It's not one of those things that has an endpoint. Okay, for those of you not familiar with the Pigeon River, it starts in western North Carolina and flows into eastern Tennessee, empties into the French Broad at Newport, which then again, of course, into the Tennessee. Our issues here that have caused part of the problem is the paper mill here at mile 63 and the power plant at the state line, mile 26. These have two of the largest impacts. Okay, so the project itself, it's a consortium of federal and state agencies, private organizations, industry, and a collaborative effort to improve an aquatic ecosystem. And here are some of our members. And if it were not for these people who volunteered from each and every one of these agencies, this Pigeon River project would not happen and would not continue to this day. I can't thank the volunteers enough. Okay, so the situation, you've got a paper mill. It began operations in 1908, okay? That was before anybody knew what an aquatic biologist was, right? Or what fish lists were. So one of the issues we had was, okay, you want to put native fish back in, well, how do we know what was in there? So fortunately, we had some folks, some experts that got together and based on other streams, and in this case, the Pigeon River, Little River. Little River is the reference stream. It is the healthiest one. And if any of you have been to Townsend, that's the Little River, okay? So they're comparing that and habitats, and they came up with an estimate of roughly 95 species of just fish, okay, that were in the Pigeon River, okay? We have thanks to Dr. David Etnar from UT and Charlie Saylor from the Tennessee Valley Authority, and I'm sure CFI contributed to this as well. And just let me introduce this. Conservation Fisheries Incorporated, Pat Rakes, Crystal, I apologize, I forgot your... Oh, still, oh, you didn't change it, okay, okay. All right, so they are here as well today. I really appreciate that. So this effluent extirpated all the snails, all the mussels, and most of the fish species. Paper mill effluents, as you can see this picture here, this is a fairly current picture. You can see it's basically this color of Coke. Okay? Back then, it was even worse. I'll show you a picture of that. But the toxics were like dioxin, chloroform, furans, and dioxins, unfortunately, are one of those pollutants that will not break down in the short term, and it'll sit in the sediment. Nasty stuff to people and critters. And then in 1930, they created a hydroelectric facility on the Pigeon. So we now have a fish barrier as well as altered flow. And that altered flow is a big deal, especially for the folks here in Tennessee. It really made a difference. One of the other things that ended up, they had to put a fish consumption advisory on the river. Now this is the second advisory. The first advisory didn't say catfish and carp. It said basically everything. Don't eat it. You know, and in this case, they said when it got better, catfish and carp were left on there because, see, they're bottom feeders. So they're more likely to get that sediment, and they're in the places where the toxins are. Yeah, so limited consumption of 1 meal a month. That's pretty bad. I would pass. Fortunately, that has since been lifted, 2007. So just to give you a shot here, this is the paper mill on one side of the bank and the wastewater treatment plant for the city of Canton. And they run them all together and it's dumped into the river. Here's a picture from back in the 80s. And the story I have from the TDEC folks is that that foam was so thick you could float a pencil on it. And the river stunk from Canton all the way to Newport. Because I always wondered why there were never buildings anywhere near the Pigeon. Now we know why. It was bad. Okay, so the first step, what happened in the early 90s is they were basically forced by a lawsuit from Tennessee to clean up their act. And this modernization process from 92 to 94 not only reduced waste, it reduced the types of toxins that were going into the waste. It also made a big difference in quantity of water. They were using 90+% of the Pigeon into their plant and then spitting it out. And so after these changes, it was down to 35%, which made a really big difference. So this led to a return of species. You know, nobody's doing anything, right? But the fish, right? They detect the changes. The water's getting better, so you had fish coming in from tributaries or from downstream, or not upstream, but downstream. And in particular, what you were getting was game fish, things that are big enough that can swim, you know, long distance. So you were still missing 24 non-game species. These are the little guys, okay? So apologize, it's just a lot of names too, but darters, minnows, and shiners is what we're talking about here. And these guys are roughly 3 to maybe 5 inches long, and they also have very short lifespans. Unlike game fish that can live for decades, these guys are lucky if they live 4 to 5 years, with the exception, and I think I want to point this one out, American brook lamprey, mountain brook lamprey. Most people don't realize we have native lampreys, unlike the nasty ones that were up in the Great Lakes. These evolved with our fish. And just to give you an idea, okay, here's a wounded darter. You can see how big he is. He fits across the palm of your hand, okay? Small, but beautiful. There's another one, look at this, blue-side darter. Again, very small. This one is, unfortunately, one of the shortest lived fish. 2 years and maybe some. Very short lifespan. So what that tells us is we have to be really careful when we're trying to reintroduce fish like that, because you got to get a whole bunch there right away, because they're going to die. So you need large numbers of fish. Okay, so in the aquatic pyramid, what we're missing are the guys in the middle there. These are primarily what the pigeon was missing at this point. And of course, we had a lot of, it was very interesting actually, we had a lot of fishermen around the Newport area that kept talking to TWRA and asking them to, hey, put some more fish in this river, we need a fishery and all that. Well, that would be fine if they had something to eat, but you know, that happened later, fortunately. Okay, and just because I wanted to show this, all right, this is a mussel right here. This is the edge of its lip. This is a display, and it's imitating a darter. And it does that, can you see that? It does that to get the fish, quite often a largemouth bass or smallmouth bass, to come by, and it will spread its glochidia onto the gills. They'll mature in anywhere from 2 weeks to a month or so, and then they drop off and become new mussels, okay? So that's the attractant. And you can't see it, but he's even got an eye spot, which is fascinating to me. So all this pollution extirpated all the snails, all the mussels, even crayfish and salamanders were no longer in the Pigeon River. There were 40 mussels approximately and 10 snail species. Okay, so like I said, the change basically took place between 92 and 94. Fish started coming back, okay? So then the question was, well, how clean is the river? How do you test that? There aren't enough test equipment to tell you, you know, how clean something is. So Steve Allstat with the USGS and Mark Fagg from TWRA decided, well, what if we took some common snails, things that you find in the other streams nearby, and put them in there and see what happens. Some tolerant ones, some of the ones that are a little hardier. So they put those in, and they survived. And so they said, okay, now we're going to try some intolerant species, and they put them in. They also survived, and while they were surviving, the first group started reproducing. So everybody started talking about that. When I say everybody, we're talking about fisheries folks, aquatics folks. We're thinking, okay, maybe it's time. So the next thought was, with the survival and reproduction, let's try to put some fish into the river. So Pigeon River Recovery Project was created in 2001. So the goal is to help restore ecosystem health by increasing aquatic biodiversity through the establishment of viable, reproducing populations of native species. Diversity. Now, I mean, look at those. Is there an aquarium fish anywhere that could compete with our own freshwater species? There aren't, in my opinion. We have just amazing fish, and most people don't even realize it, right outside their window. So then the question is, okay, we want to reestablish native fauna. How do we do that? Okay, that's a question. And fortunately, CFI had already asked that question before we started, and they gave us some good clues, some good instruction. So here are 3 of the most important criteria. Okay, so historic range, basically what that means is it was here before, okay, to the best of our knowledge, you know. And one of the keys, of course, is, well, it's in this stream right here and it's right next door. So logic says it was probably in the Pigeon, right? available habitat, sources of fish within the watershed. That's important. You want to maintain that genetic base, whatever that was, as close as possible, okay? But by far, the key is available habitat, okay? And that sometimes is very difficult to determine. Okay, so the methods we use to collect, all right, we're going to use seining, we're going to use backpack shockers. And I've got a better picture here coming up, but this is a backpack shocker. There's a battery in this compartment right here. Here's the control for voltage. It runs 23 to 25 volts basically, a half to maybe an amp. It's adjusted based on the conductivity of the water, okay? Because the higher the conductivity, in other words, the more ions and things in the water, the easier it is for the electricity to flow. So we have to turn the voltage down, because what we want to do is stun the fish. We don't want to kill them, right? Because we want to collect them and take them and release them in the pigeon. And we only use backpack shocker on those species that were just almost impossible to collect otherwise. So one of the other methods that we used a lot is called kick seining. And we collected this little guy, this blue breast darter. This is in the Nolichucky. So you get a whole bunch of volunteers, and you're basically kicking the substrate. You're literally moving the rock as you move towards that net, called a seine. Now you see several folks with buckets. That's what we'd put the fish in. So once these guys get right up to that net, they're going to pick it up. We're going to collect those fish, put them in a bucket, and then put them in holding tanks until we're done with that collection that day. Now, for some of the shiners and minnows, they don't like the fast water so much. They prefer quiet stuff. So we use, for the pool areas, we use a seine haul, okay? And we're going to pull that right up to the bank, flip it up, and get fish out of there. Same method. So here you can see a little bit better with the backpack shocking. So you can see he's got two probes here. And so the electricity is flowing between, and he's got a net on this side. And we use that because sometimes when you're actually doing the shocking part, you'll see a fish flip, go belly up, and it's starting to float outside. And it's not going to make it in the net, so he just grabs it and then tosses it in himself. And as you can see here, backpack shocking, you don't necessarily need quite as many people, which can sometimes be an issue. Okay, and then back to our little lampreys. I just wanted you to see that. We definitely have to backpack shock for lampreys. They're kind of like aquatic earthworms. They live in the substrate. Like, for example, if you're on a stream and there's an old log and you've got this big eddy pool behind it, it's real mucky, real dark dirt, right? They love that. That's their home. That's where they want to be. And we discovered, I think by accident, I think T-Deck discovered this, you put the probe over the surface of the water, literally, like where you see him holding his probes, and they either love the electricity or they hate it. We haven't figured out which, but it makes them come straight up, and then we can net them and haul them off. Okay, so collecting river snails. Very easy to do. We go to the French Broad, the Nolichucky, and just collect the common snails that are out there and transport them to the pigeon. Now, boat shocking, this is also a lot of fun. You never know what you're going to get. That's the fun part. So right here, these metal cables are where the electricity is flowing into the water. Okay? Can you see that all right? Yell at me if I block you. No. So this is going to shock them, and usually the fish are going to pop up right in this vicinity or beside the boat, and they're going to net them, bring them in. We hadn't needed to use this until the last few years, when in North Carolina we started reintroducing the red horse, a couple of species. This is a golden, also smallmouth red horse, which gets really big. And so the only way to collect these guys is by using boat shocker. Okay, back to our collection efforts. So in the stream, and we're collecting, doing kick seine operations, etc. We bring the buckets over to the staging area. So this is the staging area right along the bank in the shade. We do water exchanges to keep the water temperature cool and keep the water fresh. And quite often we'll collect more than one species, especially if we're doing shiners and that sort of thing. And so we'll split them out and put them into coolers. Here's an inside shot, some saffron shiners. And when these guys are colored up, you can see the red lips. Well, they're bright red and yellow. Just beautiful fish. Okay, so the first three fish that were going to be introduced into the pigeon were darters. Gilt darter, blue breast, and blue side darter. We needed to tag these guys. We can't tag shiners and minnows because their scales come off so easily. Just handling them is bad. They lose their scales. But darters, not so. So what we're going to do is tag them with visible implant fluorescent elastomer, which is a fancy name for medical grade silicone that's got a color. Okay? So we put the needle, so it's like the size of the needle you use for insulin, very tiny. So you put this under the skin, and you pull the needle out, injecting the elastomer, and it's just like a tattoo. This little tag has never caused an infection. We've never found a fish ever with an infection from one of these that we've recovered in the past. And he'll have that little tattoo for the rest of his life. Now, what's cool about that is that then when we go out to do the monitoring activities, and we're going to go out and see if we can find some of these fish that we've been putting in, right? When we get one, let's say I got one with a yellow tag on it. Well, I know because I tagged fish 2 years ago with a yellow tag. So I know he's been in there 2 years. So survival, right? That's telling me we definitely have survival. All right, but the more exciting one is where we go back out there and we find smaller versions of these guys, and they don't have tags. So what do we got? Reproduction. Yes, yes. And of course we want to anesthetize the fish for 2 reasons. One, because we don't want to stress the fish, so we knock them out. Second of all, have you ever tried to tag a wiggling fish? Doesn't work so good, no. And the other plus is all the VIE that we used was the fluorescent kind. So as you can see how those tags are glowing, if you use a blue light flashlight in limited light, you can still see the tags. Plus, we also discovered that when we're tagging the fish on occasion, especially if we're teaching students or somebody new how to tag, sometimes the tag will go a little too deep. And so what happens is you might only have like a spot. And so when you're out snorkeling, you might not be able to see that tag because of the skin, the scar tissue will form over it. But if you have that blue light, you can actually see it. And I only know that because I did the study in your facilities, and I had tags that disappeared, and I took that blue light and put it on it, and there it was. So it was kind of cool. Okay, so now we're going to pack for transport. So what we do is we have transport, special plastic bags. We put them back in the cooler, fill them about 1/3 full with fresh water out of the stream. We're going to put the fish into the bag, and then we're going to add oxygen to the rest of the bag. Tie it off and transport it. And they're in the cooler, and they're easily good for two hours, easy. All right, then we get to the pigeon, and we're going to acclimate them before we release them. So we float the bags because one of the most stressful things we just determined in doing our collection efforts is temperature. You have to try to keep them as close to whatever temperature they're at. So like we don't collect fish in the summer because the water temp is going to be so much cooler in most cases than the air temp that when you take them out, you're stressing them further, and we had all kinds of deaths the first time we tried to do that. So we don't do that anymore. Spring and fall, we're acclimating to temp, and then you'll notice the bag is open. Okay, what we'll do is after the temperature is pretty close, we're going to dip the edge of that plastic bag down into the water and get a little of pigeon water to mix with the water that they came in, all right? Because that's from another stream, right? So what we're doing is acclimating him now to water quality parameters like pH, conductivity, and other things, okay? So because again, that can have a shock factor. So we take our time, we get the bag full until we got, and usually they're ready to leave the bag. Now, every once in a while, I'll get one that keeps going in the back of the bag and won't leave, you know, but I guess he's missing home, you know. And then we release them, and look at that. There we got a whole bunch of gilt darters with little orange tags. Hopefully you can see that. They got little tags here on the back end. We double tag them so that when you're snorkeling, you don't always have to say, well, shoot, I can't see the right side of the fish. I don't know if he's tagged or not. So this way, we got tags on both sides, and in case the tag were to lose it. In other words, scar tissue formed over it or whatever. At least it still has one tag. We've always, and our tag retention, and I believe Pat and Crystal will agree, it's 100% almost, pretty darn close. I mean, when you tag them, they keep their tags. And so these guys are going, what in the world just happened? And that's why they're still sitting there. It's like, okay, where are we? It's Oz. Okay, so monitoring. So we're going to go back and use some of the same techniques. We're going to do seine hauls, we're going to do backpack shocking, and we're going to snorkel survey. Okay? Snorkel surveying by far is my favorite because first of all, you see fish, and it happens every time. We can go out there with that shocker and go sample this area, and we may or may not get a fish, okay, that we're looking for. right? But you go get and snorkel, guess what? They're there. They're just, they outmaneuver. Not to mention the fact.
We've seen that shot before. We're not going to get him on here.
That's right. That's right. It's like, hey, I know what that is. I'm out of here. And of course, snorkeling is just more fun, period. You get to see the entire environment. You get to see the whole community of fish as well. Okay, so the survival and relocated fish and reproduction led to the expansion from the Tennessee side of the Pigeon into North Carolina. And the reproduction was verified by Jenny Harrison when she did her master's research on the Pigeon River and looking at the migration of those recently introduced darters. She found a whole, I was amazed, an entire crowd of several dozen juvenile gilt darters. And that's what sealed it. So, and we're supported initially by just the University of Tennessee. And then after the paper mill, Blue Ridge Paper, saw the success we were having and realized that, hey, this is a good PR opportunity. They decided they would continue to support us. And the neat part is that they have supported us that entire time. And when they created, basically what happened is in 2003, they decided to create a position for the project because they wanted it to continue. Otherwise, when I got my master's, it would have been done and that would have been it. But they wanted a coordinator for the Pigeon River Recovery Project that would coordinate everything going on in the whole Pigeon, North Carolina and Tennessee. And then, now, like I said before in the beginning, what we were looking for were fish that we could find in big numbers, right? We're going to collect them and move them and put them in the Pigeon. Well, there's some on that list that you don't find. It doesn't matter where you are. You don't find them in big numbers. And tangerine darter, this beautiful fish right here, one of my favorites, or as Dr. Alford likes to say, should have been called the Tennessee darter. But anyway, CFI, Conservation Fisheries Incorporated, has not only do they propagate them, but they were the first to figure out how to propagate a species. Because think about it, there's a lot of variability when you're trying to do something like that. And Conservation Fisheries, here's some of their, this is some that they propagated to release into the Pigeon. You'll see their nice, pretty green VIE stripe, right? And they've been putting them in at the Denton site and with funding from TWRA. And there you are, Pat, at their facility. Massive tanks everywhere. They propagate for U.S. Fish and Wildlife Service, for Kentucky, for Georgia, North Carolina. They do a lot. And I just want to put in a plug for you and say, hey, that if you guys want to volunteer, guess what? They take volunteers. So here's a picture of some of the species, not all, but some of the species that have established populations, okay? But by far, the most successful has been the gilt darters. And I'm going to show you here and this is one of those things for the bird lovers. You know how the male birds are usually the pretty ones, and guess what? Fish, same thing. Okay. Now, not all, but a lot of fish, especially in the darter clan, have the males will be the really colorful one. The female, she's pretty much dark, and you can't see here real well, but the white belly, she's real pale. And of course, that's so they can hide, especially if they're, laying eggs, that kind of thing. So 16 sites in 12 miles in Tennessee, 13 sites in North Carolina. They've done really well, and what's the exciting part is that they have actually migrated on their own. It's like I was telling you, we think we find a good spot, and they go, we don't like this spot. And they migrate, and they find even better spots. And I will show you about that later here on the Tennessee side. It's quite fascinating. So here's a map just to sort of give you an idea. So all these circles are sites where fish have been reintroduced. And up here, the square boxes, and this big box, this lower section on the Pigeon in Tennessee is like the gilt darter hotspot. And none were put in here below Tannery. This is at mile 8, and they've gone almost to the mouth, almost every riffle. Just to give you an idea about the numbers we're talking about, okay, since their inception. Probably to me, the most significant is to realize that over 46,000 individual fish have been released into the Pigeon. That's a lot of fish, a lot of fish. And the point also to make is that it takes a lot to try to establish a species. You know, you're not talking a few hundred fish. You know, you really do need more than that. And the red horse are only at 575 because it's a recent reintro. We're not done with them for sure. So back to that list I had up there before, all in white. Now we got colors. Colors, basically what they're telling you is that was the year we started to reintroduce that particular or several fish, okay? That's all that is. The things with the parentheses, okay? This is where we have failed to find probably the right habitat, okay? These fish have not taken. So we've got the blue-side darter and stargazing minnow. Two things. You remember I talked about the dam and the hydroelectric facility. The hydroelectric facility is one of those that does not have a set schedule. They will throw the switch and go from 150 cubic feet per second to over 1,000. Okay, that just pretty much strips out most of the pigeon in the Tennessee side until you get down below Newport. It washes out all the sand. Well, you need sand and gravel because a lot of those small species of fish, that's what they lay eggs in. Okay, you blow it out, they don't have anything. And then even the ones that lay eggs that are scatterers, think about it. It's like they put them in this slow flowing area and they go, oh, okay, we're good. And then all of a sudden they throw the switch and it's all blown out, you know. So that's an issue. And then now we have North Carolina's list, okay? And, oh, wait a minute, let me back up for a second. So you'll see the three little plus signs. Those are the three species that have established populations. It's been very difficult on the Tennessee side. And of course, in the beginning, we thought, well, Tennessee would be the best because it's the furthest from the paper mill, right? But see, there are other things playing in here, like that hydroelectric dam. North Carolina has actually done really well. And they have definitely done better with shiners, as you can see here. And even they, though, have had problems like this saffron shiner that I showed you earlier. Apparently, they're too sensitive. So they thrive upstream of the paper mill, right in the pigeon. But when we've moved them to several different locations downstream, we never find them again. So there's something missing there. Okay, then this has been really a neat surprise, and I'm hoping it'll happen again. CFI and myself, Crystal and Pat, we went down the Pigeon snorkeling, and we actually found the river darter. I think you both found it, not me. I'm half blind. I just go along for the ride. But it was so cool. I just hope that wasn't a single occurrence. We hope that they'll be back. You know, the thing is that lower Pigeon is not real accessible, so it makes it very difficult to do monitoring. Okay, it's like you have to get in at Newport and get in a canoe and you snorkel and get in and out. You got to go the whole thing all the way to the bottom and then get out because there's no access points. But we hope they continue and come back again. Okay, now mussels, another thing we've reintroduced. This is one way to do it with this scope. The other way is to get a 5-gallon bucket and put plexiglass in the bottom. That works pretty good too. That's the cheap version. It works. And snorkeling. Okay, so what's really cool about this mussel restoration? It was again one of those things. It's like the pigeon in North Carolina had two species of mussel. The wavy-rayed lamp mussel, this picture here, and then the Appalachian elk toe. Those are the only two that are in that section because the river is much smaller, higher gradient, et cetera. But what was questionable is, well, the wavy-rayed is not listed, okay? So you can do a lot of things. They were already have a mussel hatchery where they were growing out mussels. They had lots because they were putting them in other rivers in North Carolina. So the question was, what about the pigeon? So Caroline Rooney from West Carolina University did a study where she took some and put them in a housing device, which I won't try to explain, but a silo, right? It was concrete and had a vent top and bottom and a little cage, right? So you'd put the mussels in there and then go put them in the river. The idea being that you can go find them, because think about it, what does a mussel do once you put it in the water? It burrows. So you aren't going to find them, okay? So you put them in this fancy little housing device, and you go back periodically, and you're looking for two things. A, is it alive? And B, do we have any growth? And so she did a study. Turns out she got that. In fact, the really crazy part was there was more growth on the mussels below the paper mill than above the paper mill. And what was determined was, A, the stream above the paper mill was colder, probably had less nutrients in it, whereas below the paper mill you had a higher nutrient load, right? So, and a lot more sun. The water was warmer, so you got a little more growth. But based on that, then North Carolina Wildlife Resources Commission decided it was time to put mussels into the North Carolina portion. And 4 years later, guess what? We finally found one. It was so exciting. And what's really cool though, so each one of them were tagged. They used super glue and these little plastic tags we put on them so you'd know when it went in, how big it was, all that kind of stuff. See this band right here? That's how little it was when it went in.
Oh my.
Look at all that growth. Pretty cool, huh? Yep, they're doing all right. And in Tennessee, we've had a little more trouble in terms of trying to track them down after we put them in. Now, Tennessee has a whole lot more species because it's ridge and valley, so it's lower gradient. You had a lot more species of mussels. TWRA helped. Dr. McKinney has several students that have done mussel studies, and they did the silo thing also. They did a comparison between the Pigeon and the Nolichucky. What we discovered is that, again, here's an issue for the dam. The mussels were growing more in the Nolichucky than in the Pigeon, and they believe it's because the dam is holding back all that organic matter, right? Everything drops out, and so you have less food available for the mussels. Plus, again, if you've got a big rain event, they can be generating over 2,000 cubic feet per second. So guess what? If you don't have, if you can find the substrate, if you can find the sand and gravel, it's going to get blown out. And that's what happened to 2 of the first sets of reintroductions. But they've moved downstream. They found some better spots. And then what we did is we tried, I think you see this right here, they were pit tagged. That's a positive integrated transponder. What it is it's like this little device about the size of a grain of rice. And you have a device that will read that, will pick it up. So the idea being is that if they're buried, you could find them. And we'd know, okay, so we haven't had the opportunity because the weather's been so bad this year to really get out there and do anything. We've had so many high flows, we haven't been able to look. But and the good thing is those pit tags last a long, long time. So hopefully we continue. Then there was our spiny river snail. Now this is really cool. Most common snails only live 2 to 3 years. This guy lives 15 years, okay? And that was a real blessing because they were put in initially in 2000 and no additional IOs were put in, okay? And we would go out there when we were doing snorkel surveys or even sometimes we were doing other surveys. We just look down and you'd see one of these. These are pretty good sized shells and you can see from the spikes, they kind of look like a marine snail almost. You'd see them and you go, oh, another adult, he's still alive, they're still there. And then came 2015 and we got really fortunate. What we didn't think about was just because the adults are here doesn't mean the juveniles are going to be in the same location. And that's what happened. We found them off to the sides of the stream, okay? Because think about it, they're smaller, right? They can't necessarily hang on in higher velocity water. And then again, we snorkeled again in 2016 and found even more. So we're really excited about that. Another success that we hope continues. Okay, so this is that lower Pigeon I'm talking about. So 8 miles downstream, right here, see this lowest dot? This is Tannery Island. This is the lowest point on the river that the gilt darters were released, okay? Well, guess what? We found them all the way down here. You see 54 just at site 5, which is right there. And it's interesting too, was the numbers of fish increase, what we're finding as we go downstream. Okay, so here we are in the lowest portion, okay, marginal, then we had a really great spot here at site 7. And this 9, that's a brand new site. They'd never been found there before. So they continue to migrate and find good habitat. And this is French Broad, so that's the end of the river right there. So 184. It was really exciting. And just another shot. I want you to see there underwater. There's a gilt darter right there, one right here. And you can see the substrate. What do we got? We got not just big cobble, okay, that would be this. But you've got small stuff, and that's what they need. They need the gravel and sand, et cetera. So downstream, apparently, in their opinion, is the place to go. North Carolina, okay, again, there's the 6 fish that have established populations, and you can see they're all over, lots of sites. And again, they're at sites where we never reintroduced them. So they've migrated and they're spreading. Other species. Salamanders, we had a graduate student, again, UT, and Nikki Maxwell did a salamander survey in the Pigeon, above and below the paper mill. And this is a hellbender, by the way, very sensitive species. They were found above the mill, nothing below, nothing at all. We even saw one salamander in a tiny trib, but he literally was in the trib, not in the Pigeon. It was close. Okay, so there's your full body hellbender shot, okay? There are things we still need to address. So crayfish. This was Casey Dunn's, another graduate student did a study on crayfish. They found above the mill, none below the paper mill, but we did find them on the Tennessee side. So that was a positive thing. And just for the fun of it, here's a female crayfish. That's how she carries her eggs. She tucks that tail up, takes care of them until they hatch, and then they hang around for the ride. All right, besides working on the Pigeon River project proper, okay, we've had a lot of collaboration, and we try to help with other projects, especially reintroductions, Coal Creek reintroductions. This is a small stream trib that dumps into the Clinch River. This is north of us. It's right, and it empties in right below Norris Dam. So the issue was, once upon a time, it got its name honestly. There was coal mines all around there, a lot of coal finds and things like that. But most of that is gone, and the stream got healthy. And Charlie Saylor from Tennessee Valley Authority contacted us and said, look, let's try a reintroduction here. And so one of the most successful, again, these little darters are tough critters. We put them in at 2 different sites, and we're finding them at multiple sites, the rainbow darters. They're doing really, really well. Okay. Richland Creek, this is a trib to the Pigeon in North Carolina. So we collaborated with North Carolina Wildlife Resources Commission. And their problem with Richland Creek was that someone had created a lake at near the bottom of Richland Creek. So again, blocks migration, right? Some of the issues that Richland Creek had above the lake before was they had some industrial. They also had an old waste site that seeped. Well, all that got cleaned up. They didn't have the industrial operation anymore, and it was time to put some fish in. There were 11 species that they wanted to try. And as of 2015, they actually found 11 of them. They stopped reintroductions at that point, and so they're on a break, a hiatus, basically waiting to see who does well, who doesn't, and then resurvey everything again and see. Okay, so progress. Reintroduced 27 species of fish into Tennessee and North Carolina, and as you know, not all of them are going to be winners, and some take longer than others. One of those would be our Mad Tom. 9 species of fish have recolonized 34 miles. We reintroduced 12 species of mussels at 6 sites. One thing I want to mention here about mussels, now I told you the fish have short lifespans, right? Well, the mussels can live 50 to 100 years. Wow. So for them, it's not such a big rush job, right? Because they're gonna, if they found themselves a habitat, we may not find them. We might find them for years, but all of a sudden, somebody's gonna find one if the habitat is right. That's our hope. And 3 genera of snails recolonized 15 miles in Tennessee. So it's done well. And again, I come back to our members, and I just want to mention again, Conservation Fisheries did the first reintroduction of non-game species of fish into the Great Smoky Mountains, into Abrams Creek. And all that work that you've done and are still doing similar it's just awesome. And I appreciate sharing all your knowledge and your time with us.
You've done an awesome job.
Thank you. And the volunteers again. And I will make one note here. This is Little River, where we do annual surveys on the Little River and Pigeon River, like I said, in Townsend. We had middle school kids come and volunteer and come help us. And like I said, it's one of those things where you can never have too many volunteers. Okay, so if any of you are interested, I'm sure Dr. Lippman can give you contact information. And even though I've retired, I plan on continuing to do this. And I know CFI can always, like I said, use volunteers. We'd love to see you. So, any questions? Yes?
All these species sound like a tremendous buffet for otters.
Well, this is interesting that you should say that. I can't say that we've actually seen otters. Have you ever seen any on the Pigeon? No. But that's not to say they're not there. Yeah, it doesn't mean anything because as Lynn mentioned, they're very stealth, very stealth.
Abrams Creek from being successful.
Do what now?
They've been in Abrams Creek.
Oh, and they didn't stop them. Okay. So, I mean, sure, they eat a lot of stuff, but in terms of numbers, there's a lot fewer of them than, than there are the fish. So,
Mussel populations would probably be one of the big things that would hit, and it would take a while for the mussel populations to be high enough for them to find them.
right. Yeah. And I would say this about the mussel aspect is like when the corbicula, which is the Asian clam, is a exotic that's ended up in a lot of our rivers, including Little River and even the Pigeon River. The good thing is that it provides food for the muskrats and the otters, Yes, easy to find, yeah. And it's one of my favorite things to find because I'll crush the shell on a rock, pull the meat out, and feed the fish when I'm snorkeling. They'll come right up to you and take it. It's choice food,
Even
okay?
the tangerines.
Yeah.
Tangerine darters will come right, you get this big orange fish come right up to you.
It's pretty cool, yeah. Yeah, so if nothing else, I would recommend that you ever have the opportunity, go snorkel, okay? And Little River is an awesome place to do it. Yes?
I have a couple of questions. First of all, how do you decide how many fish to remove from the river? Like, do you do any sort of like statistical analysis of like how it's going to affect the population and all that? Or
Well,
do you just kind of collect and go?
the bad news is, in the beginning, nobody had a clue, for the most part, other than recommendations. And so it was like, I want to say in the very beginning, Jonathan Burr said, let's just aim for 1,000 and see what happens. And in the case of the gilt darters, they started reproducing and we started seeing them before we got to 1,000. But yet at the same time, like the mountain mad tom that we've been trying to get established, we've already put 3,000 in, 3,000 of those. So it's one of those things where you just have to sort of, I hate to say wing it, but to a certain extent. And you don't want to keep collecting from the same spot either. Yes, that's a very valid point. It's like you don't want to decimate the existing population or you'll have nothing to go back to. And I don't know, Pat, do you guys have something that you use in terms of, okay, we can't collect at this site anymore? Do you use any kind of, or Crystal, is there anything?
We're usually dealing with much rarer fish. You got to think about that these are actually common fish where they're being collected out of.
Right.
And I mean, there are numerous studies of depletion to where you take so many fish out of a complete area and within months they're completely re-inhabited. So I would say you're probably not having much of an impact in the long term.
Yeah, yeah. I guess the best way to address it is to say, we try not to hit the same place every year. Now, in the beginning we may have, but later on we stopped doing that and we tried to find other locations. And so then we'll come back, right, to that place again. And just like Crystal said, well, there they are. It's not.
They're so mobile.
Yeah,
Some level, and
there's no problem.
you create an empty niche,
Right.
everything's going to get filled.
Right. Yeah.
I imagine that sometimes if you take individuals out of a population, it stimulates the remaining individuals to say, ooh, there's room
Right.
for more, and they reproduce more.
That's very true. That's very true. Because the little guy that's the runt's probably going to get pushed to the outer edges of what's considered good habitat.
Yeah.
Right.
Have you had any water quality problems with an upset at the paper mill that discharges more pollution than is allowed?
Oh, there's been some incidences where that's happened, and it's not just the pollutants, but it's also temperature. That's probably a big key, because they're limited. Yes, exactly, and they are supposed to be limited. Their discharge is only supposed to be 100 degrees, no more. And they had some incidences several summers ago where we were in a drought. The flows were way down. The air temperature hit I mean, it had skyrocketed, and they had an issue. We actually had a fish kill due to temperature.
I was gonna ask that.
Yeah, we had a fish kill. We did, yeah. And then,
Otherwise, there seem to be, except for that extreme, are they relatively sturdy in the water and stand upset a little bit?
yeah, it just depends on the species. You know, some are just more sensitive, and that was something that we learned was that because they did the state did a complete study of that fish kill. And they went out and counted and identified fish, and what they found, the most fish that were killed were northern hogsuckers, which is a common fish that's not threatened. It's not listed. It's not. I mean, there's usually plenty of them, but nobody realized how sensitive, and that was actually the greatest number. The other thing, though, it was like, it's one of those things where they tell you, something bad happens, but something good can come out of it. That was when we realized that the silver shiners had migrated all the way up to right below the mill because they found dead ones. It's like nobody knew that they had traveled that far that fast, So, there was a benefit there, but at the same time, it hasn't happened again. So we hope that it does not. Sure.
Okay, so the shocking
Mhm.
process.
Yes.
Are there any effects on other aquatic species, such as like the hellbender or salamanders?
Oh yeah, definitely it can be, most definitely. And one of the issues I can remember Tennessee Valley Authority, when I worked with them for a while, and we would go out in the spring, we're doing these surveys, we're using backpack shockers, and we had to skip certain streams because mussels would be reproducing, okay? And you don't want to shock when they're reproducing, okay? Because see, they're vulnerable. Like that picture I showed you of the display, they come up out of the sediment and they're exposed, and so that shocking can affect them. And unfortunately, shocking will also injure fish. It's sad but true. Sometimes, like if the fish comes in direct contact with the probe, for example, okay, that can kill them. And again, it depends, what we've got it set at. And the issue, of course, is conductivity is not uniform in the river. You're going to hit a spot that's got higher conductivity, one that's less. It's like, I wish it was more controllable, but it's not. And sometimes electricity can actually break the back of a fish. Yes?
So are you guys collecting any tissues for later genetic analysis of the fishes you're introducing? Because when you talk about seeing 7 fish, and I'm like, well, that's a heck of a bottleneck.
Yeah, right. Well, I know CFI, you guys do that on a regular basis, right? With your reintroductions. Yeah, they do fin clips all the time. We're getting ready to do that, back in 07 or 08 with the gilt darters in the Pigeon. And for whatever reason, at the time, we went from finding lots of gilt darters in the Tannery Island site to finding hardly any. So we didn't have the numbers we needed to do this, the fin clips, to do the genetic stuff. And now, of course, we can say, well, that's probably because they'd all gone downstream to the better habitat. So that's something that still needs to be done, yeah. And of course, we're not putting gilt darters still in the Pigeon. That one's completed. But
One of the problems with genetic studies is finding any funding to do
Yeah.
the genetic
Yeah.
studies.
You
Yeah.
can take the clips, but you gotta find someone to.
Well,
Pretty
I mean,
expensive,
I wonder,
yeah.
I could imagine that would be a very nice resource for later back.
Oh, yeah, definitely.
The cost of these
If
analyses
you've got
always
somebody
get
who
less.
can store it reliably.
Yeah.
long term.
And I'm sorry, you had a question?
Are you stocking with any nursery grown fish?
I guess by nursery you mean like propagated stuff? That's just for us, only the tangerine darters, okay, that CFI is doing for us. Other than that, all the fish we get, we collect from
And you just move them
one place to the other, right?
And the fish that you get from CFI that are stocked, are they as hardy as the natural?
I would say yes, in a lot of ways, but it really depends on what version of birdie you're looking at.
Yeah.
Typically, you find that captive propagated fish have less of a tendency to stress out, so you're going to have less losses whenever you actually do introduce them versus say minnows and stuff like that have been shocked and translocated and whatnot. You probably lose in about 50% of them
Right.
the next day or so.
Yeah.
So, I would say our losses are a lot less than that,
Yeah.
so.
It's, yeah. It's just like that slide that had the gilt darters that had just been released, and they were all just sitting there on the bottom in shock. It's like, hey, let's hope no big guy comes along, or he's going to have him a real feast. He's going to have a real feast. Yes.
And, yeah, speaking of the big guys coming along and eating the fish,
Mm-hmm.
do you see any effect of the bright neon
The-
strips that you use?
The tags?
The tags? I mean,
No, not really.
There've been some studies.
There were some
made in
studies.
Creek by Tennessee Tech
Oh,
students.
okay.
And they have found that, yes, some of the fish that have just been released that are tagged do get eaten.
A little
But it's
faster.
Probably within the first 24 hours.
Right.
Very, very quick. While
Very
they're still a
quick.
little bit-
When they're a little confused,
Right.
right?
We've witnessed it in
Right.
some occasions and some really small fish that we've released that
Yeah.
have been tagged.
Uh-huh.
It definitely makes them more visible.
Yeah, 'cause they're so
They're
tiny,
very
right.
cryptic species that doesn't have color on them.
Right.
But a lot of these, once they are acclimated and do get around, they're undercover and
Right.
reactive enough to where it shouldn't be too much of an issue.
And we tend
Right.
to limit tagging to fish that are cryptic, what we call cryptic. They like to hide under rocks and stuff where they're not going to be seen, where those tags aren't going to be seen. Fish that are going to, where tags are going to be a target for a predator, like a minnow, we don't tag those. It's too dangerous.
Yeah. And I,
One last question.
mhm.
Sorry, so I actually spent a lot of time on Pigeon,
Ah,
so I really like to kayak it, but
Uh-huh.
it still stinks. But apparently not as bad as it used to.
Not as bad as it used
So,
to, correct.
What is the long-term prospect for that river getting cleaner?
Well, I would venture to say that short of the paper mill closing down, I don't, I don't see that.
We lost that battle.
Yeah, yeah. So I mean, the other thing that you have to also recognize is that you're having population booms in western North Carolina like every other place. So you've got, like, I know twice we had issues where the wastewater treatment plants in some of those little communities along the Pigeon that dumped into the, they overflowed, they had this, that, and the other problems because of just so many people, you know. So you're still going to have wastewater issues, it just maybe not the, you know, the paper mill kind of problem. But I will, but one last thing is that, and because we have beefed up that part of the aquatic pyramid with all those little fish, guess what? The Pigeon has become a trophy smallmouth fishery for all of you that love to fish. I mean, and it just keeps going. I mean, it's really impressive. Yeah, it's doing well on all cycles, all circles, and the aquatic pyramid. Here's the deal, and I did not mention this, but you know, a pyramid becomes more stable the wider the base is, right? So by adding all these fish, we're making the pyramid wider. So we're making the ecosystem in the Pigeon much more stable, so that should something happen, okay, like a tanker truck ends up in the Pigeon River in the gorge, right? You know, you're gonna lose a bunch of fish. You're gonna lose a lot of this, that, and the other, but it'll come back because you've got so many different species and so much habitat that they're gonna come back, you know.
Thank you so much for your.
Yeah.